Part 3A is about what's in a cell, and then we start talking about movement of molecules. So Brownian motion, we see it in lab. Molecules, matter is always in motion. They're wiggling around thanks to kinetic energy. Now, this is not the same as cellular energy, ATP. Okay, matter is always in motion, has some kinetic energy in it, molecules are hitting each other. And through this kinetic energy, they can move by simple diffusion or osmosis or facilitated diffusion / filtration. Simple diffusion is moving from high concentration to low concentration, no membrane to go through, so perfume. You get Giorgio perfume. It's very expensive because it's highly concentrated in smell good molecules. You spray it, it's going to make it to the other side of the room. When a person wears it and walks through the hallway, you're going to be able to smell that person when that person's long gone. The Walmart brand, they might be able to capture the essence of the Giorgio smell, but it's going to be a lot less concentrated in those smell good molecules. Hence the cost. It's cheaper because it doesn't have as much stuff in it. It's a lot more water. So since it's less concentrated, it's not going to make it as far. When you smell it, it's a fairly high concentration, and maybe move to the person next to them, potentially the person 2 people away. Osmosis is water moving from water's high concentration to water's lower concentration through a semi-permeable membrane. So it's a type of diffusion, but it's specifically the molecule that we're talking about moving is water. And then facilitated diffusion, we're still talking about things moving, but there's a driving force. So the driving force in lab was gravity. In the body, it's going to be blood pressure. So it's almost like we have something that's forcing the move, some sort of outside pressure that's forcing the move. In lab, we had a filter, and then we had some dirt and sand and rocks, and we had starch, sugar. Then we poured some distilled water into that, and we watched gravity pull it through the filter. The filter was keeping big things in, but little things were getting to go through. This sometimes is what happens at our cell membranes. The little things can get in, sometimes, still rules, but big things cannot get into the cell, and vice versa. So now we're going to talk about active membrane transport, and there's 2 kinds. One of them is going to involve those proteins that are embedded inside of the cell membrane. The other is going to be where we're going to make a vesicle, also known as a vacuole. We're going to make one of those, and it's going to move either in or out of the cell. Both are going to require ATP, and both, there's a reason that we're going to have to use this type of transport instead of passive transport. So things that are too big to get through those protein channels, they're going to need help. Things that are not hydrophobic, so things that are hydrophilic, are going to need help. Sometimes if it's charged, that's not on here, but I digress. And then sometimes maybe things need to move up its concentration gradient, not down. So in other words, instead of moving from high concentration to low concentration, it needs to move from low concentration to an already crowded area. And we have reasons for that. It has to do most of the time with that charge that I've mentioned that you're going to have on your cell membrane. Your cell membranes have a charge. On the inside is the most important one, but we also have a charge on the outside. So we're still going to use these carrier proteins if we're just talking about moving through the cell membrane without a vesicle. And sometimes it's going to be more than one thing passing through this, this integral protein, this channel. Sometimes, with the help of ATP, we're going to have one thing moving in while another thing moves out. That's an antiporter. Sometimes we're going to have things that move in together. So two things with the help of ATP get to come into the cell, or two things get to go out of the cell. A lot of times we're talking about moving against the concentration gradient. So if we're trying to force something to go from low concentration to an already crowded situation, high concentration, we're going to need some cellular energy for that movement. This is a picture of symporter and antiporter, showing symporter, just like Nemo and the sea anemone have a symbiotic relationship. They work together. A symporter moves things together, and an antiporter moves so you have one molecule moving in while the other molecule moves in the opposite direction. So an antagonistic movement. Now we're going to have another thing I'm going to introduce to you. Two general categories are active transport. In active transport are primary and secondary. So in primary active transport, the direct energy comes from ATP breaking into ADP. The best example we have of this is the sodium-potassium pump. There's others, but the sodium-potassium pump is going to be the first one that we are going to introduce. Secondary active transport means that because something happened at a different pump, such as some sort of ATPase pump, there's going to be energy stored in concentration at the gradient that makes another pump work. And this is most common with symporters and antiporters. So sometimes it's going to be a true ATP-based pump, thanks to an enzyme called ATPase. But sometimes it's a pump that got its energy from somewhere else, thanks to a gradient created by ions. The sodium-potassium pump has two asterisks here. That's because it's really, 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 really important. So this pump is a protein, and he's an enzyme, specifically ATPase, and his job is to move sodium and potassium against each other. Now here's the thing. You have to memorize sodium is highest in the concentration outside of a cell, potassium is highest concentration inside of the cell. And movement of these two ions actually will trigger some cells to do their job, such as your neurons. Your neurons are going to fire, which means they're going to send their information of importance. Or your muscles are going to fire, they're going to contract, thanks to movement of sodium and potassium. This is a picture depicting the pump. So these enzymes are ATPase. They're ATPase enzyme. Enzymes are proteins. And the hexagons are sodium, right here. The ovals are potassium. Here we're in the extracellular space. Here we're in the cytoplasm. And what's going to happen, so I just told you that sodium is highest in concentration on the outside of cells. Looky here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 versus 1, 2, 3. Sodium highest on the outside of a cell. Memorize that. Now, I'm telling you that potassium is highest in concentration on the inside of the cell, so the ovals. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. And then on the outside of the cell, 1, 2, 3. Even if you want to count this, 4 or 5. Clearly more potassium on the inside of the cell. What's going to happen with the with the sodium potassium pump? We're going to move 3 sodiums out. So by cleaving off a phosphate group off of ATP, making ADP, we're going to make that pump start up. And when it starts up, we're going to push 3 sodiums into an already crowded situation, and we're going to push, so like this right here, and we're going to push 2 potassiums into an already crowded cytoplasm full of potassiums. That's what the pump does. This has to do with that charge that's on our cell membranes. We do have little leak channels. So if you remember from 3A, we have some channels that stay open all the time. And so because of the sodium that is being allowed slowly into the cell and potassium that's allowed slowly out of the cell, we have to pump it all the time to make it as highest of a concentration as possible, because we need to maintain the inside of the cell membrane. So we're inside the cell, right at the bottom of the phospholipid bilayer. We have to maintain this, negative 70 millivolts, in order to excite tissues that need to get excited. So the purpose of this pump is to maintain this electrochemical gradient of negative 70 millivolts.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on 2 factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
Okay, secondary active transport is when the energy is coming from another site. So let's say like the ATPase, that's the sodium potassium pump. It creates a gradient by moving ions in high concentration on one side, and then the secondary guy can actually drag another molecule in with him, for example, or go out, can be out. Picture. Okay, vesicular transport, we're going to move some bigger things. And I've actually already kind of alluded to this when I talked about there's a certain white blood cell that likes to eat bacteria. The way that he eats it is he actually engulfs it and creates a vesicle, a vacuole, with the bacteria in it. So vesicular transport is going to be moving large particles where it actually has to genuinely eat it or drink something. And it's going to be called vesicles. A vesicle is the same thing as a vacuole. It's going to be stored till later. So that cell is going to have to use some ATP to kind of open up its mouth, if you will, to drink or to eat something that is on the outside of it. Or some cells make things that need to leave the cell. And so whatever it makes and packages up in its little vesicle / vacuole, it's going to leave the cell through that little vesicle, and that requires ATP also. So when a cell actually takes something in, the term is endocytosis. So cyto, cell, endo, inside. And there's 3 specific kinds of endocytosis. I've alluded to this already too, because in lab we did a pinocytic vesicle. So a pinocytic vesicle is when that cell has drank something. Pino, to drink. Phago is to eat. So phagocytosis is when the cell has eaten something solid. But I haven't mentioned receptor-mediated endocytosis. We'll do that one in a minute. The term exocytosis is when something leaves the cell. This vacuole that is inside the cell needs to go out of the cell. I'm going to skip transcytosis and vesicular trafficking for another AP2 class. This picture is actually a genuine example of something that I would do in my own work. Dog's got an ear infection. I need to be able to pick the right medicine to fix that ear. So I'm going to do cytology. I'm going to swab that ear. I'm going to roll it onto a slide. I'm going to stain that slide and see what I see. I don't like this slide. I mean, this dog's ear could be worse, but this is actually showing different kinds of white blood cells right here. That's white blood cell. This is a white blood cell right here. And what's on the inside of this white cell, these little things like this, that's bacteria. So this is probably a staph because the way it's bundling up in sets of 2, sometimes they bundle up in sets of 4, but whatever. This white blood cell, called a neutrophil, he is going to phagocytize bacteria. He is going to actually use his phospholipid bilayer membrane and wrap that membrane around that bacteria and through ATP bring it into the cell. We have other white blood cells that do this besides a neutrophil, but that's what's on this slide. Phagocytosis, to eat and bring into the cell. So phagocytosis is a type of endocytosis. Pinocytosis, pino to drink. So this is when maybe a cell is actually going to drink in some water. Maybe there's some solutes in there like sodium chloride, but drinking in some fluid. This is how some nutrients are actually absorbed in the small intestines. Okay, receptor-mediated endocytosis. We're going to have some substances that act like a lock and key mechanism that are actually going to bind on a protein, so a specific, very specific receptor, and that tells the cell, hey, I'm here. I'm one of those things that you want to take into the cell. So go ahead and create that vesicle and drag me in. On this, you can see this cell membrane is starting to dip in like this as we get these lock and key mechanisms going on here to here. And it's going to end up pinching this area off and bringing this part right here together with this part right here, creating this vesicle. Do, like that. Exocytosis is the opposite. So the cell has made this little vacuole or vesicle, needs to leave. Cells that make hormones, they need to be exocytosed. They're going to, let's do insulin. Insulin was made at the pancreas, and an endocrine gland does not have a duct. Exocrine glands use a duct. Endocrine glands don't use a duct. So we've made some hormones. Let's say pancreas made some insulin. It is going to package it up. Insulin's a protein. Package it up into a nice little vacuole until your pancreas gets the message that you have a lot of glucose floating around. You just had 5 Snickers bars or whatever, a meal. And then your pancreas got that message, and so the insulin is going to exocytose out of those pancreatic cells, eventually getting into the blood and sending out the message, you need to let the sugar in. This happens with other things too, listed: neurotransmitters, mucus, cellular waste. This is a picture of exocytosis. This is a cute little video right here. Well, cute, that's in the eyes of the beholder, but anyway, this is some sort of gland that has made something, and it is exocytosing out of the cell. Vesicles are also going to be made of a phospholipid bilayer. So you have this resting membrane potential that I told you about. On the inside of the cell, it's going to be a negative 70 millivolts, and potassium is the key player in maintaining the resting membrane potential. Because we have the inside of the cell at negative 70, we have the outside of the cell something else. Remember, it's loaded with sodium. Sodium is positively charged, and so the charges on the outside of a cell versus the inside of the cell are different. So that means that it's polarized. You've heard the term polar opposites, right? So that cell membrane is polarized. Here we go. Put this in your memory banks forever and ever and ever. I've already mentioned it again. It must be important. Sodium is highest in concentration on the outside of the cell. Potassium is highest in concentration on the inside of the cell. We have a pump that's going to push even more sodium out and bring even more potassium in. So going from low concentration to an already high concentration requires ATP. This is necessary for especially neurons and muscle cells to do their job. When they do their job, charges change. This is an example of that receptor mediated. This is a clinical view, so this is a disruption, so not homeostatic balanced. It's genetic. And what happens is some people So low density lipoprotein is the bad cholesterol, LDL. HDL is good cholesterol, LDL is bad cholesterol. And what happens is, especially found in like meats and such, you ate a high fatty meal. Let's say you just love to eat all the fat off the edge of your steak or your pork chop or whatever. That's chock full of low density lipoproteins. Well, the cell still wants to go ahead and bring that into the cell. It's going to try and figure out what to do with it. So the LDL has a place to bind on a cell, LDL receptors. Well, if you are born with poorly functioning or completely absent LDL receptors, you can't get LDL into your cells. So therefore, it stays floating around in the liquid part of your blood that you know as serum or plasma. And that's going to build up plaque. And it's bad enough that if you have this disease, you have potential to die in your teens of a heart attack because of the blockages that it causes. Your watery liquid part of your blood starts to become chock full of fat, and it just starts to clog up the inside of the vessels, just like really hard water does to your plumbing. Why is she on my slide? I hate spiders. I really do. But she is an excellent example. If you don't know what she is a black widow. She's an excellent example of somebody that breaks the rules. So what she does, her venom actually pokes holes in the cell membrane. She destroys the rules. And as she destroys those rules, one of the things that we don't want going in and out willy-nilly of a cell, is especially a neuron, is calcium. We want calcium to come in to trigger a nervous action potential to make the neuron fire, but there has to be a slow level of influx, and there has to be a reason that there is an influx. So because the black widow's poison pokes holes in the phospholipid bilayer, calcium comes rushing into the cell, and then we get symptoms of poisoning.